cholesterol metabolism. We're going to break down the entire life story of this molecule, and you'll see why it's so critical for both the human body and for your board exams. Let's get right into it. You know, the whole story of cholesterol is really a paradox. On one hand, it's absolutely essential for us to live, but on the other, it's public enemy number one when it comes to heart disease. So the big question we're going to tackle is this: why is our body's most essential fat also one of its most notorious killers? Getting a handle on this double-edged sword is absolutely key. So to figure that out, we're basically going to follow cholesterol's entire life story. First, we'll see why it's so vital. Then we'll take a tour of the synthesis factory where it's built from scratch. After that, we'll track its two major road trips through the body, one starting from the food we eat and the other from the liver. Then we'll meet the cleanup crew. And finally, we'll wrap it all up with a super high-yield summary of everything you have to know.
Okay, so first things first. Why do we even have cholesterol floating around? Before we start calling it good or bad, you have to understand that without it, well, we just wouldn't be here. Simple as that.
Yeah, so cholesterol is definitely not the enemy we sometimes make it out to be. Its job is incredibly important. Think about it. It gives every single cell membrane in your body the right amount of fluidity. It's a starting molecule for all steroid hormones, like cortisol, testosterone, estrogen. The liver uses it to cook up bile acids so you can digest fats, and your skin needs it to make vitamin D from the sun. The bottom line? Our body needs it. Period.
Now, because it's so critical, the body is way too smart to just rely on your diet. It has its own in-house production plant, and the main headquarters for this factory is the liver. It's responsible for making about 70% of all the cholesterol in your body. The whole process is basically an assembly line. It all starts with a simple building block, acetyl-CoA. A couple of those get linked together, they go through a few modifications, and they form this key intermediate called HMG-CoA. Then comes the most important, the most regulated step of the whole pathway. HMG-CoA gets turned into mavolinate. And from that point on, a whole cascade of reactions transforms mavolinate into our final product, cholesterol. Okay, let's pause and zoom in on that one middle step. The enzyme that does the job of converting HMG-CoA to mavolinate is called HMG-CoA reductase. This is the master switch. It's the rate-limiting step, which means the speed of this one reaction dictates the output of the entire cholesterol factory. If you're going to remember one enzyme from this whole pathway for your exams, this is the one. And this is precisely why it's so clinically important. The most powerful cholesterol-lowering drugs on the planet, statins, they do one thing and one thing only. They block HMG-CoA reductase. It's like they drop a massive roadblock right in the middle of that assembly line, which dramatically cuts down how much cholesterol the body can actually make. Okay, so that covers how we make our own cholesterol. But what happens to the cholesterol and fats we get from, say, a cheeseburger? That brings us to the body's incredible lipid transport system, and we'll start with the exogenous pathway. Exogenous just means from the outside. So after you eat a fatty meal, all that fat gets absorbed by your intestines. But here's the problem. Fat and blood are like oil and water. They just don't mix. So the intestinal cells have to get clever. They package all those lipids, triglycerides, cholesterol, the works, into these special delivery vehicles. The best way to think of them as giant transport trucks, and they're called chylomicrons. Let's follow one of these trucks on its journey. It gets assembled with a unique protein license plate called ApoB48, and then it pulls out into the bloodstream. As it drives around, it uses another protein, ApoC2, to basically honk its horn at an enzyme on blood vessel walls called lipoprotein lipase, or LPL. Think of LPL as the unloading crew. It jumps into action, breaks down the triglycerides inside the chylomicron, and delivers those fatty acids to your muscles and fat tissue. Now, after dropping off most of its cargo, the truck is pretty much empty. This is what we call a chylomicron remnant. It now uses a different protein, ApoE, which you can think of as its return to sender shipping label, to get recognized by the liver. The liver then pulls it out of circulation, and the journey for that dietary fat is officially over. Right. So that handles the fats from our food. But what about all that cholesterol and fat that the liver factory was just busy making? That needs its own distribution network. And that's where the endogenous pathway comes in. So the liver bundles up its own homemade triglycerides and cholesterol into a different kind of truck. This one is called VLDL, which stands for very low-density lipoprotein, and it gets a different license plate, a really important one, ApoB100. This marker is the key that lets it deliver its cargo to cells all over the body. So, this VLDL truck leaves the liver.
And, just like its cousin the chylomicron, it uses ApoC2 to activate LPL and start unloading triglycerides. As it loses all that cargo, it physically shrinks and gets denser. It morphs first into something called IDL, or intermediate density lipoprotein, and then finally into LDL, or low-density lipoprotein.
And right here is where we meet the famous bad cholesterol. LDL's main job is actually super important. It's the final delivery service for cholesterol itself. It cruises through the blood, docks onto LDL receptors on cells everywhere, and delivers the cholesterol they need for all those vital functions we talked about. The reason it gets a bad rap, is when you have too much LDL. It can start dumping its cholesterol cargo into artery walls, and that's what leads to dangerous plaque buildup.
Okay, so we've got delivery trucks coming from the gut and from the liver. But any good city needs a waste management system, right? A cleanup crew? What happens to all the extra cholesterol hanging around in our tissues? That's where our final and maybe most important pathway comes in: reverse cholesterol transport.
And this job belongs to HDL, high-density lipoprotein, the good cholesterol. The best way to think of HDL is as the body's recycling truck. Its entire mission is to drive around to all the neighborhoods, all our tissues, scavenge for any excess cholesterol that's been left lying around, and haul it back to the liver to be safely disposed of or recycled.
So how does it actually do this? Well, HDL has a special tool on its surface, a protein called ApoAI. This protein flips the switch on an enzyme called LCAT. And what LCAT does is it chemically modifies the cholesterol that HDL picks up, which effectively traps it inside the HDL particle. This makes sure none of the cholesterol falls off the truck during its return trip to the liver.
Whew. Okay, we have covered a lot of ground. So let's pull everything together into the most important high-yield summary for your exams. These are the absolute must-know players in the entire story of cholesterol.
You've got to know this table cold. ApoA1, think A for activating LCAT, found on HDL. ApoC48 is exclusive to chylomicrons from the gut. ApoC100 is the big one, on VLDL and LDL, and it's what binds to the LDL receptor. ApoC2, think C for cofactor. It's the universal activator for LPL, our cargo unloading enzyme. And finally, ApoE, think E for end. It marks the remnant particles for their end, getting taken up by the liver. And this slide right here, this puts it all together. You can see the whole system in one view. You can trace the exogenous pathway, starting with those chylomicrons from the intestine. Then you can follow the endogenous pathway, watching the liver send out VLDL, which becomes LDL to deliver cholesterol. And finally, you can see the reverse transport pathway, with HDL as the cleanup crew, bringing it all back home to the liver. It's a really beautiful dynamic system when it's working right.
So we'll end with a thought to take with you. We've just mapped out the perfectly healthy, normal flow of cholesterol. The really crucial next step for you is to start asking, what happens when any one of these finely tuned steps breaks down? What if the LDL receptor is defective? What if LPL doesn't work? Because understanding this perfect system, that's your foundation for understanding the pathology of lipid disorders.